So I think we can start. And hello, everyone. And it's a pleasure to welcome you to the third webinar organized by the Data and Partnership Department of Unicancer. This webinar focuses on the topic real-world data in oncology, uses and new perspective. And will be hosted by three prestigious speakers: Gulben Tedze, Head of Partnership, Data and Innovation at Unicancer; Thomas Fideron, Head of Biostatistics and Health Data Science at Oncopole Claudius Regaud; and Louise Bachet, co-founder and Head of the Advanced Methods Department at Oriana. There will be 40 minutes of presentation, followed by 20 minutes for questions at the end of presentation. So thank you for your participation, and let's start.
Yeah, thank you, Daniela, for the presentation, and hi, hello, everybody. So I'm delighted to present to you how real-world data is driving innovation in oncology, and to provide you with an overview and to elucidate why real-world data is accelerating innovation. I'm going to walk you through the landscape. So you see here, from the from the left to the right of the slide, we observe several waves of therapeutic innovation, spanning from chemotherapy, targeted kinase inhibitor, antibody, and so on. And with this different wave of innovation, a Spanish study demonstrated that from 1999 to 2016, innovation has increased patient survival by 2.7 years. And so this dynamic of innovation has significantly enhanced patient outcomes, so confirming tremendous impact of innovation on patient care. Hence, that's why we believe that innovation improves patient care and real-world data as well. However, to ensure a successful development, company must address challenges. On the next slide, I will outline the challenges that need to be tackled to secure therapeutic asset development. And as you know, you can see 4 challenges, including identifying right target, conducting relevant clinical trials, understanding epidemiology, and ensuring the scalability of results. And a failure to address these challenges leads to negative impact. That means that gaps in data, in order to fulfill this challenge, if there's no data, it impedes the development of a drug. And for example, a wrong target lead to unspecific adverse reaction, and in the same manner, irrelevant clinical trial leads to loss of success rate, and a paucity of data leads to epidemiology. Paucity data in epidemiology leads to restricted label, and a poor scalability of result leads to low clinical interest, especially for health technology assessment. Therefore, I'm going to show you how real-world data bridge these gaps and can be leveraged to secure a development process. So you can see on this slide from left to the right that real-world data elicit mechanism of a disease, so it permits to really refine target. The data can be leveraged to refine clinical trial design. And as real-world data fills a crucial evidence gap in epidemiology with patient journey treatment patterns, and real-world data secures scalability of results. And Thomas and Louise are going to show you how real-world data are used in order to restore a comparison. So it means with a single arm clinical trial, how real-world data are used to have the relevant comparison. Therefore, it shows that real-world data is bridging this gap, and consequently, real-world data supports the innovation, and in the end, it improves care for patients. So now let's delve into Unicancer programs. So what are the current unicancer programs? So our flagship programs are ESME and CANTO, and it address major evidence gap in breast and lung cancer. And for ESME, it's a high granularity database which allows outcome research, and with 200 parameters, it's possible to have outcomes like overall survival and progression-free survival. And for CANTO, it's a deep cohort with a blood sample, with a genetic information, and with patient-reported results, which enable a complex analysis. And the roadmap of a unicancer in real-world data involve enabling automatic data collection to achieve a better contemporaneity. So we have a program which name is Oncodata Hub, ODH, which enables automatic data collection, and we want to scale up to other indication as well. To go the extra mile and level up the scientific value proposition, Unicancer is launching a linkage with National Health Insurance Database to have information regarding ambulatory care and health economics data. So I show you in this slide our 4 main programs. You can read several scientific use cases that are addressed with this program. And you can see that ESME has been accredited by French HTA as a reliable source of data in order to complete market access dossier. And so in this slide, I show you few examples of publication concerning a valorization of a real data program. One with the external control arm on amivantamab, and Thomas is going to present you this scientific work. Moreover, the research work on decision-making for entrectinib is going to be presented by Louise as well, on other publications as target simulation trials, cost effectiveness research on international comparison. So how to run a partnership with a unicancer? So partner need to design a first study concept and share a synopsis. And we have a scientific committee who defines data collection, scope of work, and research objectives. This committee provides independent validation of a research project. It meets every 2 months that ensure a timely decision making. So there is an update of the full database, which is run every year with a strict quality control. Analysis are run with updated data, providing expected results with a new version of the database every year, and deliverable includes a publication as well. As demonstrated on this slide, we have several industry partnerships, and our data have been utilized for HTA, and so it means health technology assessment, and international regulatory submission purposes. Thus, it shows the trust of the industry players and confirm that unicancer databases are a reliable source for evidence generation. So you have our mail in this slide. So if you have additional questions, we will be happy to discuss it after the webinar. So thank you for your attention. And now I hand over the floor to Thomas Filleron for his presentation. And thank you. Have a great webinar.
Thank you very much, Gulben. Louise, can you share the slide? So in this second part of the webinar, I'll go to focus on a publication realized in Advanced Therapeutics by Troy et al. This publication relied on adjusted treatment comparison of amivantamab versus real clinical practice for patients with advanced non-small cell lung cancer with EGFR exon 20 insertion mutation. in the first time, I'll go to present the medical context. Lung cancer is the leading cause of cancer death for men and women worldwide. There were more than 2.2 million new cases of lung cancer in 2020. Non-small cell lung cancer is the most common type of lung cancer, affecting 85% of patients with lung cancer. Over the past 10 years, non-small cell lung cancer has become the archetype for precision cancer medicine. Molecular testing of lung adenocarcinoma for oncogenic driver mutations has become standard in pathologic practice. EGFR mutations are common in non-small cell lung cancer. Approximately 30% of non-small cell lung tumors harbor a mutation in the EGFR gene. Prevalence ranges from 14% in Europe and 38% in Asia. The majority of EGFR mutations comprise exon 19 deletions and exon 21 substitutions. There exists also uncommon mutation. One of them is exon 20 insertion mutations. Across Europe and US, the frequency of exon 20 insertion mutation range from 4% to 12% of all EGFR mutations. The frequency of EGFR exon 20 insertions ranges from 0.3% to 2.6% of all non-small cell lung cancer cases. This patient population have a poorer prognosis compared with patients with common EGFR mutation. Until recently, there was a lack of specific and effective treatment for patients with advanced EGFR mutated non-small cell lung cancer with exon 20 insertions following platinum-based therapy at second line or later. Moreover, there's no treatment guidelines exist for these specific patient populations. Chrysalis is the first in human phase 1 dose escalation and dose expansion study. The trial evaluates the efficacy, safety of amivantamab in patients with advanced non-small cell lung cancer. During the dose expansion phase, activity was investigated in different cohorts. The primary endpoint of the dose expansion phase was overall response rate. Key secondary endpoints are duration of response, clinical benefit rate, progression-free survival, and overall survival. The cohort denoted D+, correspond to the EGFR exon 20 insertion population with previous treatment with platinum-based chemotherapy. 81 patients were considered in the pivotal efficacy population. The objective response rate was estimated to 13% and the clinical benefit rate was rated to 74%. During the conduct of the trial, amivantamab received breakthrough therapy designation for the EGFR exon 20 insertion population who had previous treatment with platinum-based chemotherapy. This regulatory approval was based on single-arm trial evidence. After publication of the activity results, conducting a randomized clinical trial was not a feasible option. As regard to the severity of the disease and response rate observed in the trial, there was a lack of equipoise. Moreover, EGFR mutation exon 20 insertion are rare. Identifying this mutation via conventional PCR method is challenging. So, it was proposed to conduct individual patient data-based adjustment treatment comparison of amivantamab versus real-world clinical practice. The primary objective is to compare the efficacy of amivantamab as assessed in the Chrysalis trial to real-world clinical practice from Europe and the US in patients with advanced EGFR mutated non-small cell lung cancer with exon 20 insertion following platinum-based therapy at second line or later. The Chrysalis cohort used in this comparison correspond to cohort D+. This corresponds to patients with EGFR exon 20 insertion who had progressed on or after prior platinum-based chemotherapy. 114 patients had received their first dose of amivantamab. The real-world evidence portion of the analysis is based on previously collected data. The real-world data sources used to provide comparative data in the analysis were from US and Europe. For US, there were 3 sources of data. For Europe, there was 4 sources of data. One of them is ESME. A direct access to IPD sources was available for all sources except ESME database. different endpoints to compare patients from Chrysalis patients from the external data sources. The same inclusion and exclusion criteria were applied. Some eligibility criteria were common to all real-world data sources, and others were specific. In the main analysis, Chrysalis was compared to a pooled EU and US real-world clinical practice cohort. This base case analysis was composed by 401 treatment lines. Sensitivity analysis explore comparison between Chrysalis and a pooled EU real-world cohort and between Chrysalis and a pooled US cohort. Different endpoints were evaluated in the analysis. Common endpoints to all real-world clinical practice data were overall survival and time to next treatment. Progression-free survival and objective response rate were not available from all sources. In the results section, I'll go to present only overall survival results. Rarely, the treated and control populations can be assumed similar. To account for differences in patient population between Chrysalis and the real-world data sources, adjustment was carried out. Potential confounders were identified by a systematic literature review, clinical expert opinion, and data availability. 7 key variables were identified. Adjustment was conducted separately for data sources according to direct access to the data. Real-world data sources with direct access were pooled. All common variables across Chrysalis and the databases were included in the adjustment. This corresponds to 4 key variables. For ESME, data were balanced versus Chrysalis independent of other data sources. All key common variables across Chrysalis and ESME were included in the adjustment. Propensity score methods were used to mimic the effect of randomization by creating a balance between 2 treatment groups with respect to important baseline covariates. The inverse probability weighting approach and additive weighting scheme was then used to generate a comparative arm. This table presents baseline patients and disease characteristics for Chrysalis and real-world data with direct access. Baseline characteristics were comparable prior to adjustment. However, the rate of patients with brain metastasis was slightly higher in the real-world data sources than in the Chrysalis trial. For ESME, data differ substantially from the Chrysalis data. After adjustment, balanced diagnoses were conducted to determine if the propensity score weighting has removed observed systematic differences between Chrysalis and real-world clinical practice. For data sources with direct access, comparability was improved. As example, the rate of patients with brain metastasis was not similar. For ESME, similarity between the dataset was improved, but some difference remains. Data with direct access and reconstructed IPW ESME data were pooled. Balanced diagnoses were conducted to determine if observed systematic differences between Chrysalis and real-world data pool were removed. There is a good overlap between propensity score distributions between the Chrysalis and the real-world cohort. The standard Z-mean difference after ATT weighting became closer to 0. As balanced diagnostics were good, efficacy analyses were conducted. This slide presents overall survival results. Overall survival was in blue for amivantamab. Survival rate for the real-world data pre- and post-adjustment were similar. Median OS in the ATT-adjusted population was estimated to 12.5 months. For the comparison, pre- and post-adjustment hazard ratio were similar. 0.45 for the pre-adjustment comparison and 0.47 for the post-adjustment comparison. As highlighted in these presentations, the use of real-world data to complement or replace randomized clinical trials when they are not feasible is of great interest. This is particularly true to leverage limitations from single-arm study. But their use may present biases, potential confounding, and pitfalls. Robust process may help to minimize biases. This process would include considerations of data quality, data comprehensiveness and completeness, and comparability to potential experimental arm. If there are major issues with the validity or accuracy of the data, Statistical methods cannot salvage the comparison. Finally, robustness assessment as sensitivity analysis, quantitative bias analysis need to be conducted. I let you the floor, Louise.
Thank you, Thomas. So I'm going to explain to illustrate the use of real-world data with another example, the use case called ROSLIC, starting by the medical context, guidelines, and clinical trials, and then I will explain the design of the ROSLIC study, which is an indirect treatment comparison study on the French ROS1 plus NSCLC patients. And we will finish with some key messages and food for thought. So, as mentioned by Thomas, NSCLC is an aggressive disease that represents a significant burden for the patients. Lung cancer is associated with significant impairment in quality of life that is reported to be higher than with other malignancies. Another kind of mutations is a ROS1 plus disease that accounts for a small proportion of lung cancer cases, but many patients present with advanced stage disease. The central nervous system metastases are common in advanced ROS1 plus NSCLC and present a significant burden for patients. In this context, entrectinib treatment was developed. Entrectinib is an oral potential and selective ROS1 TRK ALK tyrosine kinase inhibitor that is CNS active. And during the clinical development of entrectinib, 5 clinical trials were conducted, mostly single-arm trial. And in 3 of them, there were subgroup of patients corresponding to adult patients with locally advanced or metastatic NSCLC, the trial LK, STARTREK1, and STARTREK2. So these subgroups of patients from the 3 clinical trials were used to support entrectinib for dossier reimbursement along with indirect treatment comparisons. In our case, this study then is an indirect treatment comparison study for French ROS1 plus metastatic NSCLC patients with the partnership of Unicancer and Roche, conducted by IQVIA and Oriana. And it answered to the need to have comparative data on comparators recognized by the French Health Technology Assessment Haute Autorité de Santé, HAS, and in this case, mainly chemotherapies. So we need to have comparative data on ROS1 plus metastatic NSCLC on French patients. Then the design of the study, the primary objective is an indirect comparison of the primary outcome, progression-free survival, on patients treated in real-life setting with recognized French Health Technology Assessment comparators versus entrectinib. In the first line, and then in the second line, for the patients corresponding to the population of interest, so ROS1 plus metastatic and NSCLC, and the statistical methods that was used was the matching adjusted indirect comparison, which is a statistical method like a propensity score that is similar to a propensity score weighting process, adapted when the data of individual patient data in one group is available, but only aggregated data in the other group are available. This is a second data use of 2 groups, 2 arms, The first arm is the combined aggregated data of the 3 clinical trials I just mentioned before, composed by the subpopulation of ROS1 plus metastatic NSCLC patients treated with entrectinib in LK, Star Trek 1 and Star Trek 2. This is the experimental arm. And this experimental arm is compared to the individual patient data from a French cohort, the subgroup of patients of ROS1 plus metastatic patients in the ESME cohort mentioned by Gulven in introduction during 3 years. This is the comparator arm. So the study populations are in the entrectinib arm composed of 124 patients, 60 in the first line and 64 in the second line. And in the ESME database, the recognized French HTA comparator is composed of 60 patients, 30 in first line and 30 in second line. In first line, it's mostly chemotherapy alone or in combinations with or without bevacizumab. And in second line, it's mostly chemotherapy alone or in association, and also a ROS1 inhibitor, crizotinib. We faced many challenges. The first point is data comparability and quality. The second challenge is the missing values that is common when we use real-world data. And the third challenge is small sample size due to the rarity of the mutation. And because the arms are not randomized, we need to account for confounding bias. Another challenge is that it's a secondary use of data. So we want to avoid data dredging, and we need to have a priori methodology that is defined before knowing the outcomes to avoid data dredging, so that the methodology is not defined by the results. And one difficulty also is that the data of entrectinib are not, the individual patient data for entrectinib are not available, and we only have aggregated data. And other challenges that I'm not going to focus on today. So the methodology that we used was a 3-step methodology. After the design of the study, during the design of the study, we defined the covariates that we need to take into account. So the covariate selection during the protocol writing based on literature review and expert opinion. Just as a reminder, the objective of the MAIC is to realign populations here between the aggregated data and the individual patient data on these covariates. The first step then is the feasibility assessment. The comparison of the study design, eligibility criteria, baseline characteristics, and outcome definitions between entrectinib studies and the ESME cohort. In our case here, the feasibility assessment was positive. So the second step was the estimation of the weights for the comparator arm. Modeling the weights estimation was done using logistic regression with the method of moments for the statisticians that are listening today. In addition, multiple imputations were used to allow the inclusion of major prognostic factors in the weighting process. In this case, ECOG status, with nearly half of the patients in the ECOG cohort with missing values and also smoking status with 2 patients with missing data. The question is how to obtain a satisfactory model for the estimation of weights with these 50 imputed data sets in a small sample size settings, so facing some convergence problem. We predefine steps for selecting the best model that calculates the weights for each imputed data set in a small sample size setting with multiple imputation. It also provides, this methodology defined a priori will provide a rating grid of the number of actions that were necessary to reach the final model without data dredging. And then the final step is the comparison of outcomes. So the outcome results and sensitivity analysis bias. Here we use the weighted Cox model for the hazard ratio estimation, and then the Rabinus rule for outcome aggregation and computation of p-values, etc. Several sensitivity analyses, planned and post-hoc, were conducted to evaluate the robustness of the results to the assumptions. For these methods to be better accepted, we have developed a rigorous, transparent, and reproducible methodology. Such qualities are more necessary as these analyses are conducted on real-world data with unbalanced arms and the presence of missing data, and in a rare disease context with small sample size. If you want more details, you can see this poster that we presented during a Congress 2 years ago. What is ongoing now? I'm not showing the final results of this study because it's still ongoing and the publications will be done very soon, I hope. Is the quantitative bias analysis as sensitivity analysis. We will start with the bias plots on unmeasured confounding. And here is an example from a previous publication. The e-value is a statistical measure introduced to quantify the minimum strength of association that an unmeasured confounder would need to have with both the exposure and the outcome to fully explain away the observed association. In other words, it represents the minimum level of confounding that would be required to nullify the observed effect. As illustrated here, an e-value of 3 and 3.331 mean that to nullify the effect of entrectinib versus the French HTA, meaning an hazard ratio of 1 on the disease progression, and unmeasured binary covariates would need to have an adjusted related risk with the outcome of 3.3 and an adjusted relative risk with the exposure of 3.3. The idea is to see how our results are robust to the assumption that the unmeasured confounding can't change the conclusion. A second sensitivity analysis is performed on missing data, and it is possible to see the impacts of the assumption of the missing at random using this kind of tipping point analysis, which is a sensitivity analysis that helps to assess the robustness of the results to departure from missing at random assumption by modifying the imputations to partially missing variables towards missing not at random. So to run scenarios where the missing values are correlated with the outcome. The tipping point analysis in this context of missing data refers to determining the level of missingness of correlation with outcomes at which the study findings become significantly compromised or biased, and then to assess robustness of results to missing data. In our case, we had to adapt these concepts of quantitative bias assessment with matching adjusted indirect comparison, multiple imputations, etc., program of convergence. So it was not so easy. So to finish, some key messages. This illustration with the Roslick use case show that it is possible to compare aggregated clinical trials versus real-world standard of care in the French setting with some strengths and weaknesses. We have to confess that the perfect indirect comparison does not exist, but the methodology that we suggest has strengths in numbers that I show that we can assess the uncertainty due to missing data and unmeasured factors. So this is possible if we select the good cohorts in France, in Europe, in US, for their data quality. So to be able to use the same inclusion-exclusion criteria in the real-world data. In our case, to select patients with the ROS1+ mutation and with robust data. And then by applying adequate methodology. Thank you very much for your attention. And we will be very pleased to answer to your question.
Yes, so thank you for your presentation. And yes, now it's time for a question. You can write your question in the chat, so do not hesitate. But I think we have already 2 questions from Lister Poon. The first one is for Gulven. Gulven mentioned that you perform annual database updates with quality control. What metrics do you use to define quality? And how do you measure these quality metrics?
Yeah, so thank you for the question. It's a comparison with a dossier. So for example, for ESME, it's a collection which is performed by a CRA, for example, in each site on experts. The data are structured in the ESME database, and so we take by random a sample, for example, like 5% of dossier, and we compare with the source dossier if there is a good level of comparability of the results. So that's the level of quality check we use to ensure data quality on ESME.
Okay, so thank you, Gulven. And another question from Lister Poon again. This time is for Thomas. Have you done a follow-up study on real-world results of amivantamab? Does it match the Chrysalis trials?
Thanks for your question, Lister. To my knowledge, we don't have a follow-up study on the real-world results of amivantamab for ESME, but I have no information concerning other database.
Okay, and don't hesitate to write questions. Otherwise, yes, we have 1 question. I'm curious to get your thought on synthetic data versus real-world data. What are possible use cases where synthetic data is better than real-world data? And where are they complementary?
Thank you for your question. Very interesting. It's only a thought. In our cases, we only spoke about rare disease, rare mutations. So where the simulation of data to have synthetic data is not easy to have a validated model of simulation. So in the cases we presented, I'm not sure that synthetic data would be very robust. But maybe in other cases, I think that synthetic data could be complementary. But I'm not a specialist, and I don't want to.
Yeah, it's a very good question because there is like a two-way to have this kind of synthetic data. You can have a mechanistic model, which is much more close to the disease. So you have a modelization, for example, of the metastases, how they circulate in the bloodstream, and the level of biomarkers that you have on certain cells, and how the mechanism of action is currently acting on these cells. On there is like two publications which has been made with this capacity to have information before the publication. So it allows like to have a simulation of a trial with this mechanistic approach. And for synthetic simulated data with like a data model, we are I think at the beginning of this used, but we can envision for example the capability to have micro simulation, so simulation with a very large cohort. So it means it's much more power for certain hypotheses. Or to be able to have like extended follow-up, which can be simulated. So it's a state-of-the-art use of this data. And I think new publication will be, I think, available soon. But you have these two ways of use simulation from a mechanistic model or a disease model, much more data-driven.
I agree, Gudven, with your last point, mostly how to predict long-term results.
Yeah, and there is another question. Have you done work with digital twins? Does the methods give similar results to synthetic control arms?
With we never work on digital twins. Sorry.
But this is a topic very interesting, and I'm sure that use cases, success story, are going to be published and see in the future the use of digital twins.
For sure, it's close to a simulation question. At this level, the statistical approach, it's like counterfactual. So it's like the same patient with other kind of condition. We restore this objective to have like a twins, like the same patient with the propensity score. So we do that with historical data. So that's historical data in order to have a patient which is the most similar to the patient we treated. But yeah, I don't know,
Mm.
Thomas and Louise, how you see, because historical data allows to make this comparison very close with alleviating bias. And how do you see like digital twins? Because I feel that it's a lot of historical data which is used in order to have a comparison with either a single control arm or for hybrid clinical trial.
Thomas, do you want to answer?
I search answer.
That's complementary. Today, we have a good feedback of the use of synthetic control arm based on historical data. But we don't know how the digital twins are going to be accepted. So I think that it can be very useful during clinical development to anticipate, to optimize the clinical trial design.
Yeah, I think it's a level of trust of the data, because historical data, it exists. And if we use simulated data, I don't know how it can be assessed by HTA. So for instance, it's like a use of this high level of confidence data, because it's like trusted data. For example, that's a reason why ESME is accredited by a French health technology assessment body. It's because they have a very strong trust of how data are retrieved and controlled. And I guess there is like a discussion to have with HTA on the regulator in order to use simulated data on a digital twin as well. But I guess because it is accelerating innovation, it can be a very good tool in order to bridge some gaps that are not fulfilled by a current real-world data program.
So thank you, Lister Boone. And we have another question for Aurélien Belot. What are, in your opinion, the main statistical challenges when conducting this study using real-world data and aiming for indirect comparison?
Thanks for your question, Aurélien. I think there is a lot of challenge. First, for me, the first challenge is first to define the estimate reflecting the clinical questions. Second, to rely on the adequate selection of external controls from previous randomized clinical trials or real-world data. And finally, to choose the optimal statistical approach to estimate the target treatment effect according to the estimate. But I think one major challenge is to have data, good quality data. Without data, we may not have a good statistical analysis.
The challenges that are highlighted by the recent guidelines published by the EMA, FDA, and also HAS are first, as you said, Thomas, to define it, to predefine the question, and to predefine all the questions, then the inclusion exclusion criteria, etc. And also what you mentioned, Thomas, but maybe another challenge, two other challenges are the comparability of the definition of outcomes. because the two arms are from two separate settings, clinical trials, compared to real-world data. So to be sure that the definitions are aligned between the two data sources. And also, as I tried to illustrate, it's how we assess the robustness of the results to the several assumptions for imputation of missing data, unmeasured confounders, etc.
Yes, I'm totally agree with it. It's necessary to add a bias analysis as bias cannot be totally eliminated.
Okay, so thank you for your question. Are there other questions for the last 5 minutes? Okay, so I have a general question. How we can access to unicancer database, for example?
Yeah, so thank you for your question. So it's a partnership. So as mentioned, the committee, the scientific committee is managing all the requests on the data. So it's synopsis, which is presented to the scientific committee, and the scientific committee evaluate it in order to run the project. And after all the analysis as are performed on the warehouse of Unicancer, so it means that it's an in-house process. So it's a partnership between usually industry and Unicancer in order to address a scientific question. So that's so it means that data stay in the Unicancer data warehouse. And for head-to-head comparison, it means that we need to receive the data on our server, for example, for the Chrysalis study, and we perform the data-to-data comparison within our server with the scientific analysis plan.
Okay, so thank you for your answer. And I will check if there are other questions. Not for the moment. So if you want to say other things, or if it's okay for you, we can stop.
Thank you.
So thank you for all the participants. And we are glad to let you know maybe the next webinar that will be organized. And thank you again for your participation, and thank you for the speakers. It was very interesting, and have a good night. Thank you.
Thank you. Good day.
Bye-bye.
See you.
Goodbye.
Bye-bye.